All right, everybody, we're going to go ahead and move on to chapter 19. I mentioned this at the very end of our last lecture, but remember, chapter 19 has two different slide decks. So obviously, we have a lot of differences in the male and female reproductive system. So we separated those completely. And these first few slides that we're going to go over is considered kind of our intro material. So some of it will apply to both systems. So that's what we'll start with. And I'll kind of let you know when we're going to talk specifically about the male system. All right. As a species, of course, we rely on our reproductive system to continue generation after generation. So the species could not survive without that ability to reproduce. But if we removed all of our reproductive system organs, we would still be able to survive as a human being. So, for example, our last system we talked about was the urinary system. We said if we got rid of our urinary system or the kidneys, specifically the filters, then none of our other systems could compensate for what they do and we could not survive. Now, none of our other systems obviously can do the functioning job of the reproductive system, but we can remove that system and still live and function as individuals. So I just want to clear up this first bullet. And of course, the reproductive system is essential in development of kind of structural and functional differences. So not just anatomy, but physiology between the male and female sex. It's going to influence human behavior because a lot of the hormones that influence this system, and of course, produce offspring. So three out of our four functions here are going to go with both systems. So one, two, and four, we can count for both. And then number three, development and nourishment of a new individual is only going to coincide with the female system because she is equipped with the uterus to be able to carry an embryo and fetus. Production of gametes occurs in both systems. So gamete is a sex cell. In males, that's going to be the sperm. In females, that's going to be an egg cell. Or you'll see in our next slide deck, another word for that is an oocyte. Fertilization is really kind of both systems are responsible for that because we need a sperm and an egg to meet for fertilization. We said development and nourishment of a new individual is only accomplished by the female system. And we'll talk about multiple reproductive hormones. And we'll talk about the different roles these hormones have within our two different systems, male versus female. So the actual formation of gametes, the formation of our sex cells. So these are going to be our reproductive cells. Like we said, that was going to be a sperm in males, an oocyte or an egg cell in female. So that's the kind of official name for an egg cell. And so it's really important to understand this last bullet here. So our gametes only have half the total number of chromosomes as a somatic cell. So we've seen this word soma or somatic multiple times. Anytime you see somatic, you know we're referring to our kind of normal body cells. In this case, our gametes are going to be considered haploid cells. So haploid means half. So we have 23 total chromosomes within a gamete. A full set of DNA in a somatic cell is going to have 46 total chromosomes or 23 pairs. So when you see this number 23, you need to first look whether it says 23 pairs of chromosomes or just 23 chromosomes, because that's going to be very different answers. And it makes sense that we're only going to have 23 chromosomes in a gamete because if we need 46 total for a full set, a full recipe, then we're going to get 23 from the sperm, 23 from the oocyte, and then when they combine to fertilize, that's going to be our kind of full recipe, 46 chromosomes. So if we started with 23 pairs in our gametes like our somatic cells, when fertilization occurred, we now have twice as much DNA as we should. So that's why we only have 23 total chromosomes. And in chapter 20, we'll touch on chromosomes again. So you'll be able to see a little bit of an illustration of that. This word meiosis is also very important to remember. And so meiosis is the special type of cell division that our gametes go through to end up as haploid cells with only 23 chromosomes. And so mitosis, we talked about briefly way back in Unit 1, and that is kind of our normal somatic cell division. So we start with one cell that has a full recipe in mitosis, and we get two cells that have a full recipe. So that's for somatic. That's mitosis with a T. In meiosis, we're going to have one cell that starts with a full recipe, and that's actually going to divide multiple times to end up with four cells that each contain 23 chromosomes. So we end up with half the number of chromosomes, but twice the number of cells as an end result compared to meiosis. Compared to mitosis. All right, so puberty in general is kind of classified or characterized is probably a better word by the beginning of the release of GnRH. So we mentioned this in our endocrine chapter. That's gonadotropin releasing hormone. And I like to kind of describe this as the first domino in puberty. And so before the onset of puberty, so in females, that's usually somewhere between 11 and 13. In males, it's typically a little later, probably more like 13 to 15. And the reason why we don't develop some of our secondary sex characteristics and the other things that go along with puberty is because we have an inhibition or a suppression of this GnRH. We don't have much in the system. That first domino has not dropped yet. And so when puberty begins, our hypothalamus, so remember that's all the way up in the brain, that's we've got all of our releasing hormones. The hypothalamus is no longer going to be sensitive to that inhibition. And so, because it's no longer being inhibited, it's going to release GnRH. And this happens in what we describe a pulse-like manner. So, we get kind of waves of GnRH that start to flood our system. And so, we said GnRH is the first domino, and it's going to knock over two new dominoes. So once GnRH is in the system, that's going to result in LH, which is luteinizing hormone, and FSH, follicle stimulating hormone, to be released. Like I said, you can think of it as a domino effect because it goes GnRH and then LH and FH kind of knock over together, and that's going to continue the domino effect of our different sex hormones. This pattern is going to continue all the way until kind of puberty is fully achieved. And not even, I shouldn't even say puberty is complete, but this whole hormone interaction has been achieved. So, really, these hormones are going to come into play much after puberty ends. So, it's not like these hormones stop once we get to our early to mid 20s. This is going to continue well into adulthood. And we'll talk about that a little bit more as well. So I'm not going to go more into detail on the function of LH and FSH right now because we're going to start talking about the different systems, and these hormones have different roles in our different systems. So just kind of keep those on the back burner, keep them in the back of your mind. Okay, this is a slide that you guys should definitely know. So fertilization, we mentioned as one of our primary functions, is when we have a union of a sperm and an oocyte. And so the zygote is what we call a fertilized oocyte. And so this is after the oocyte is fertilized and is going to begin to divide. So from a zygote, sometimes you may see another word called blastocyst that is kind of in between a zygote and embryo. But for the purposes of our class, we say that the zygote is going to then develop into an embryo within 3 to 14 days. And so that kind of sounds like probably a weird window, but it's based on partly implantation as well as how many cells are present, how much division has happened. So we can't really give a definite number on our transition from zygote to embryo. Embryo and fetus are not interchangeable. So sometimes I think kind of out in the news or when you're seeing stuff not necessarily in an anatomy and physiology context, the word embryo and fetus kind of get interchanged. but really it's an embryo up until week 8. So not all the way, about a month before the first trimester will end. And then from an embryo, we have a fetus. So the fetus is going to be from 8 weeks all the way up to birth. So just make sure you know we start as a zygote. That's the fertilized oocyte. Remember, it's an embryo until week 8. And then week 8 on, we have a fetus. All right, so this is where we're going to go into kind of only the male system for now. So the intro stuff, those first few slides kind of apply across the board. Now we're going to talk about the male specifically. As always, I'm going to obviously mention kind of everything that we cover in our chapter, but I will do my best to point out the really need to know stuff and some of the nice to know stuff so that you guys can hone in for your studying. So our male reproductive system, you can see everything labeled here. The testes are our primary sex organ. We'll talk a little bit more about these in a few slides. And other than the testes, where we're actually producing sperm, we have a series of ducts and accessory glands, which are that go along with some other supporting structures that all are going to kind of help the male reproductive system accomplish its end goal of delivering sperm to the female reproductive system. And so we'll go through all these different ducts, accessory glands, and supporting structures as we go through our slides here. So that last slide was a picture obviously zoomed out. This is much more zoomed in. So we're really looking at just the pelvic cavity here. If we took a cross section, kind of split the pelvis in half and looked in. And so this is a good slide to refer back to after we've gone through all of the processes that we're going to talk about because it contains all of our structures. So this is a good one to use to study from because it doesn't have any of the other information. It just has your actual anatomy labeled. So you can kind of apply some of the physiology and processes we talk about and kind of visualize how that's going to occur. All right, another way to view it, this is a cross section where we're really looking in from the front of the body. And so this is another one that has everything labeled. There's a lot more labels here than you guys really need to know. So as always, make sure you're paying attention to this stuff that is bolded, but more importantly, really pay attention to this stuff that we have actual words and I talk about a little bit more. That's going to be what you need to key in on. And this image you should definitely study because you may or may not see one of these two images on your exam. All right, one of our first supportive structures is the scrotum. And so that's actually going to contain the testes. And the reason that we need to have the scrotum so that the testes are not directly inside of the body is that sperm has to develop at temperatures less than normal body temperature. And so we actually have two different muscles, the dartos muscle and the cremaster muscle, and they're kind of muscles that surround the scrotum. Our cremaster muscles are actually extensions of our abdominal muscles, and you can contract these to pull the testes closer to the body. So we've talked about the word homeostasis and how the body always likes to maintain a perfect environment. Same thing goes for our testes and sperm development. So we're trying to maintain a temperature that's just a couple degrees lower than our body temperature. So if our kind of body temperature is running hotter, if we're in a hotter environment, we can relax these muscles and the testes will be farther away from the body. If we're in a colder environment, we can contract these muscles and pull the bodies closer to the testes. Typically in response to cold. But another thing to point out here is that our skeletal muscle, this cremaster muscle, if we get into that fight or flight response, our sympathetic system is amped up, we'll actually activate this muscle to pull the testes closer to the body as well. But really think about it from a temperature standpoint in terms of the scrotum and that muscle action, because we need to keep the sperm developing at the perfect temperature. So our gonad, we said, remember, gonadotropin releasing hormone was the first domino. So the gonad is the primary sex organ. For the males, the gonad is the testes. So it produces sperm, it produces testosterone. We just said they're located in the scrotum, and they contain these structures called seminiferous tubules, and that's where our sperm is going to begin to be produced. And we're going to talk about the pathway that sperm goes through. Okay, the seminiferous tubules are kind of located within the testes and organized into these lobules, we call them. And so within these tubes, we have interstitial cells. They're going to secrete testosterone. That's our primary male sex hormone. We have germ cells. Those are kind of like our baby sperm cells. So they're not sperm yet, but that's what is going to eventually become sperm cells. And then we have sustentacular cells that nourish the germ cells and also produce some hormones to help with development. So I always think sustentacular, think sustain. Their job is to sustain the germ cells so that they can become sperm. Okay, we're not going to go super in depth on meiosis, but you should at least know that the term spermatogenesis is what we kind of term the process of forming the male gamete. You should also know that in males, this is going to begin at puberty, and this is going to continue throughout the lifespan. So technically, if you have kind of all the hormones that are still cycling properly and at the proper levels, males can produce viable sperm their whole life. Males produce about 90 million sperm a day, which seems like a large number, obviously, but when we talk about ejaculation, you'll see that a lot of sperm exit in one ejaculation. So there's a reason we produce so many million. And so you kind of constantly have different amounts of sperm at different stages of development. So you have some that are just developing, and you have some that are mature. In the grand scheme, not that this number is important to you guys, but it takes 64 to 72 days for sperm cells to fully mature, go through the whole process. And so we're not going to get in depth with this. Like I said, you need to know that meiosis is this special type of cell division, but this kind of just walks you through briefly what happens. So we said that we start with one germ cell that kind of has full recipe. That goes through mitosis. So that ends up with two germ cells. We end up going through two different steps of meiosis, and then we end up with four spermatozoa that each have 23 total chromosomes. Now we're super zoomed in because we're looking at one individual sperm cell. So the head of our sperm cell is going to contain the nucleus. So that's very important because that's got the DNA that has the recipe. It has this area called the acrosome. You can see it's kind of pointed, and it also contains enzymes in the actual acrosome portion of the head that is going to help the sperm cell break through the membrane of the egg during fertilization. M for middle or mid piece, M for mitochondria. So that's going to make ATP. So that's the powerhouse. And the tail we call a flagellum, and that's how our sperm are actually going to be able to move or swim. All right, we're going to get into the different duct systems that we have, and this is kind of how we get sperm from the testes through the urethra. That's kind of what I'm going to focus on as we go through this pathway. So the epididymis, this is a cross section. This is the epididymis in our other picture. And so this is where our seminiferous tubules empty the new sperm. And so they're going to do most of their maturation in the testes. And then once they get to a certain point of maturation, they migrate to the epididymis. And that's where they're going to finish maturing and then be stored until ejaculation. So all of our sperm is going to be housed within the epididymis prior to the whole process of ejaculation. Our next stop is the ductus deferens. A lot of times you'll see that called the vas deferens. And it's this long tube. It goes up over the bladder and it runs down into the seminal vesicle and eventually the ejaculatory duct. Kind of fun fact, not going to be tested on this, but something clinical that you can know is that this is actually what is cut during a vasectomy. Because in a sense, our vas deferens is really just a carrier, a tunnel that the sperm can travel through to get to the seminal vesicle and ejaculatory duct. So if we disconnect that tunnel from mature sperm to the ejaculatory duct, there's no way for viable sperm to get to the semen. Okay, our ejaculatory duct is next. And so the seminal vesicle is also in there. We won't skip that, but we'll come back to it. Yeah, I didn't miss it. Just making sure. Okay, so the seminal vesicle is going to contribute some secretions as well. The sperm we said is traveling through the vas deferens, and that's going to join in the ejaculatory duct. So you can see this prostate gland here again. We talked about this in our chapter 18 with the male. So that ejaculatory duct travels directly through the prostate to meet with the urethra. And so, like we talked about in our urinary chapter, if you're getting enlargement of the prostate and constriction of these pathways, that can cause some issues with blockage. And then the end goal, in a sense, is to get to the urethra because that's how our semen is going to get to the external environment. So you see the ejaculatory duct is here. Our urethra extends from the bladder here. So that's where they're going to meet. And so our urethra is really named in three different regions. You have the prostatic region, which is going to be the portion that's actually in the prostate. You have the membranous region where it's kind of going from prostate to the penis. And then the longest portion is the spongy urethra. Sometimes you'll see the spongy urethra called the penile urethra because it's the portion that's actually in the penis. So our urethra in males is the passageway for our reproductive fluids, the semen, as well as urine, but not at the same time. And kind of to really simplify it and make it easy, when semen is going to enter the urethra from the ejaculatory duct, we have to close off the internal urethral sphincter that's controlling bladder release so no urine can enter. When we let that relax to let urine enter the urethra, we have to shut off the gate here so that semen won't enter. So they can't enter at the same time. All right, so the penis is the male copulatory organ. So that means it is involved in the act of intercourse. It's delivering or transferring sperm from male to female. So the primary sex organ for males is the testes. The copulatory organ is the penis. So don't get those two mixed up. We just mentioned, of course, it also excretes urine because it contains the urethra. The actual distal end or the head of the penis is known as the glans penis. And then the actual opening is officially termed as the external urethral orifice or external urethral opening. Okay, we're not going to get super in depth with the whole sexual process, but we are going to touch on it because obviously it's a very important process within our reproductive system to actually achieve its function. So, erectile tissue is kind of the first step. So, we actually have three different columns, we term them, of erectile tissue that really just fill with blood. So, we have two that we call the corpora cavernosa, and we have one called the corpus spongiosum. And really what you guys should take from this is that for an erection to be achieved, these three columns all have to vasodilate, or the blood cells within them will vasodilate, and they will result in an erection because vasodilation means wider arteries and it means a lot more blood flow into that tissue. All right, we're going to touch on some of the glands that also contribute secretions to the semen. So the first one is the prostate gland. We said it surrounds both the ejaculatory duct and the urethra, and it contributes enzymes that actually help to liquefy semen further. So in a sense, it's making it easier for the semen to be delivered inside the female reproductive tract. One of the other pieces it contributes is a buffer or a basic solution to help neutralize the acidity of the vagina. So we'll talk about this in the female system chapter, but the vagina is more acidic. It has a lower pH than our body. And remember, it's an opening to the external environment. So that's part of its protective mechanism to prevent unwanted microorganisms from entering the body. Two very small glands called the bulbourethral glands also contribute secretions. They're located close to the prostate gland. And so they do not contribute secretions until we kind of go through the process of sexual stimulation. So it helps to lubricate the glans penis, this head of the penis, and it also contributes to neutralizing acidity. So we mentioned in the prostate gland that we're trying to protect against the acidity of the vagina, but our urine is also very acidic. That's going to travel through the urethra, the same pathway. So we need to protect it against any kind of leftover urine or leftover acidity within the urethra itself. And then last but not least, we have the seminal vesicles. And so this is going to contribute a majority of our secretion to semen. And so they join with the ejaculatory duct. So that was vas deferens to ejaculatory duct via the seminal vesicle. And its main job is to provide fructose, just a different type of sugar. We need sugar for ATP production. So it adds fructose that the sperm are able to utilize, send to their mitochondria to produce ATP so that they can actually swim to their destination. It also contains a substance known as prostaglandins. And so that helps to decrease mucus thickness around the cervix and the uterine tube. So we'll see that fertilization actually takes place in the uterine or fallopian tubes. So this is, again, trying to make it easier for our semen and really the sperm to be able to move through the reproductive tract and get to their end destination. It also contains some coagulants that help to deliver semen to our female system. And so this is the big one that you should remember in terms of what it's contributing. It's got to contribute the energy. Without the fructose from the seminal vesicle, our sperm would not have the energy to get to their destination. So semen itself is not just sperm, it's a mixture of all of those secretions from the different glands and the sperm itself that we just talked about. And so we kind of have said this a couple times. I've been a little repetitive in the fact that all of these secretions are to aid the sperm in reaching the egg cell. So that's the goal. I said a majority of fluid is coming from the seminal vesicles. A good portion is also going to come from the prostate gland, only a little bit from our bulbourethral glands, and really only a little bit from the testes. So only a small percentage of that fluid is really the sperm itself. And so earlier we said that male systems make 90 million sperm a day, and 2 to 5 milliliters of semen are going to be ejaculated at once, meaning 200 to 500 million sperm are going to leave the male system during ejaculation. So that's why they're producing 90 million a day, because we're going to get rid of 2 to 5 days worth of sperm production in one kind of average ejaculation. Sperm can live for 72 hours once inside the female reproductive tract. Kind of put that in the back of your mind for right now. We'll kind of talk about how the egg cell functions during fertilization in that process, and so we'll hit this number again. So just keep that in the back of your mind for now. Okay, and so this is a great study slide because you should definitely know this order. And this is where I was saying kind of studying something like this and then going back to just your picture slide and talking yourself through it is a great way to check your understanding. We can't see the seminiferous tubules because remember, they're located within the testes. So if we took a cross section, we could see all those lobules and tubules within. And so that's where, one, we're going to produce testosterone, and two, we're going to produce our sperm cells. Remember, they mature most of the way there and then head to the epididymis to finish maturing, and they are stored in the epididymis until ejaculation. We're going to travel up the ductus deferens or vas deferens, and that is going to meet with the ejaculatory duct. So we're not necessarily skipping the seminal vesicle. The seminal vesicle is contributing secretions, so it's really adding to it. It's not necessarily a stop or a step. It's going to add to our sperm that's coming from the epididymis, and they're going to empty into the ejaculatory duct. Then you have all three portions of the urethra next. So the ejaculatory duct meets with the prostatic urethra. We have the membranous urethra and then the spongy urethra. And to exit, they will exit through the external urethral orifice. So you guys should know this path. All right, here's our first few dominoes from the beginning of the lecture. So we're going to talk about these specific to the male system now. And so we said GnRH, gonadotropin releasing hormone, is the first domino that's going to knock down our next two dominoes together, LH and FSH. So in the male system, LH, or luteinizing hormone, is going to stimulate secretion of the testosterone. So without LH, we're not going to secrete testosterone. So no testosterone, no sperm production. FSH, follicle stimulating hormone, is going to prompt spermatogenesis. So that's that process of going from one sperm with a full recipe, one germ cell, all the way down to 4 spermatozoa. So LH for testosterone in the male system, FSH for spermatogenesis. And just a reminder, this we kind of tested more in chapter 10, but both of these, LH and FSH, are coming from the anterior pituitary. So we had a whole bunch of hormones coming from there that had a variety of different functions. But just remember, these are coming from the anterior pituitary. And then GnRH, we said, was a releasing hormone, so we know that's from the hypothalamus. One hormone that we haven't talked about yet, but contributes to the feedback loop and kind of balance of our hormone system, is inhibin. And so this also comes from the seminiferous tubules. And if we kind of don't need to bump up how much sperm we're producing, then we can secrete inhibin to kind of slow or stop that process. So that stops FSH from prompting more spermatogenesis. All right, we've said this a few times, but here's a slide for you to go over it. Testosterone is our primary or major male sex hormone. We already saw that it's produced by the interstitial cells of the seminiferous tubules, and it's involved in both the development and maintenance of our reproductive system, and especially the testes themselves. It also is responsible for secondary sex characteristics. So these are the characteristics that develop because of the onset of puberty. So stuff like tougher skin texture, we get fat distribution changes. We'll talk about that a little more in the female system. We have hair growth in the genital region, the armpits, more on the body. Skeletal muscle will grow even if you're not necessarily working out. You're going to get an increase in skeletal muscle, and we'll get changes within the larynx, within the voice box. So we all know kind of the classic voice crack or deeper voice that goes along with puberty in males. Okay, we're going to go through the sexual process here. So we talked about the erectile tissue earlier. So that kind of starts with neural input that results in those vasodilation of our 3 columns. Blood is going to fill that tissue. We then get parasympathetic action potentials that are going to, or really parasympathetic action potentials are responsible for that process. We're then going to have blood filling something called our sinusoids. So sinusoids are veins in this case, but they're really very permeable capillaries. So the erectile tissue is going to compress these sinuses or sinusoids, and that's going to prevent blood flow back out of the penis so the erection can be maintained. The next stop, or part of the process is what we call emission, and this is when we get an actual accumulation of sperm cells and semen within the urethra. So as this sexual process starts and the erection is obtained, that is when we're going to start the kind of travel of sperm from epididymis up through the vas deferens to the ejaculatory duct. This portion is stimulated by sympathetic action potentials. So erection was parasympathetic, emission is sympathetic. And so this is going to result in all those ducts that we talked about to contract and start to release secretions, as well as the seminal vesicle, the bulbourethral gland, the prostate gland, all those accessory glands we discussed. And this is when semen's going to begin to accumulate in the urethra. So we said we call that emission. And then the last step is the actual ejaculation. And so that is when we're going to have a forceful expulsion of those secretions, the semen that has built up within the urethra. We get a contraction of the smooth muscle internal urethral sphincter. That's going to prevent urine from entering the urethra and prevent semen from going back up into the bladder. We again have sympathetic nerve contributions, and that is going to cause rhythmic contractions that result in ejaculation. So ejaculation is also accompanied by intense pleasure, increased heart rate, and blood pressure because some of the changes that are going on in the brain at that point, and we term that climax or orgasm. This is something we include because we'll point out the difference in the female system, but we have resolution and refractory periods in the male system. So after ejaculation, we get both muscular and psychological relaxation, and they term that resolution. So after this resolution period, or as this resolution period is occurring, we're getting, again, sympathetic nerves are going to contribute to cause the arteries and arterioles to constrict. This is going to reduce that blood flow. So the sympathetic part isn't as important to you guys, but kind of think it's the reverse of what happened to achieve an erection. So we had parasympathetic input that causes vasodilation, that erectile tissue fills with blood. After ejaculation, now we're going to get sympathetic nerves telling that those blood vessels to constrict again, and the penis will become flaccid. The male system also has something known as the refractory period, and the male is going to be unable to achieve another orgasm until for some people a few minutes, some people a few hours. And as we get older, this refractory period tends to lengthen. So just kind of keep this in mind again. We're not going to test you on this in depth by any means, but there is a little bit of a difference in our female system that we'll talk about in terms of resolution and refractory. So keep this in mind. All right, guys, thanks for listening, and I will hop over to the next slide deck and we'll talk about the female reproductive system.